big welcome to Helena Oakey and Dr. Wook-Kim coming to us from Australia to discuss a nested case control study for ENDIA BIOS. Helena Oakey, PhD, has been a senior biostatistician on the environmental determinants of islet autoimmunity, otherwise known as ENDIA, the team based at the University of Adelaide. And Wook-Kim is also a PhD, has been characterizing human viromics, characterizing the population of all human infectious viruses to identify causal infectious agents in undiagnosed infectious disease cases and elucidating the role of viral infections in chronic disease development. He's been doing application of cutting-edge virome characterization methods, including methods based on next generation sequencing or NGS, such as virome capture sequencing, VARCAP-seq, Amplicon WGS, and VerScan, which is related to pan-viral serology. And they're going to speak to us about, again, this protocol development for a nested case control study design for omics investigations in ENDIA. Thank you both for joining us. It's morning time in Australia, and really appreciate you fitting us into your day.
Oh, thanks for inviting us to talk today. So I'm Helena Oakey. I'm the ENDIA statistician. I'm part of the data management team and a steering committee member. My statistical research interests include experimental design, linear mixed models, and of course, statistical omics analysis.
Yeah, I mean, we're very lucky to have Helena on the team. Thanks for inviting me. I'm Wook. This is actually my only slide for the talk, and then I'll chime in the discussion or questions. But I'm really here to, as a user's perspective, I guess the protocol that Helena's developed, which she'll go through, is very much applicable to all the omics themes. And in ENDIA, I lead the virology theme, so I'm responsible for looking at characterizing the gut virome, respiratory virome, and the blood virome using antiviral antibody screening, as well as looking at actual just parent-reported history of infections. So that's what we're coordinating here at the moment for the nested case control group, which consists of the first 54 cases in ENDIA of islet autoimmunity. So I'm also a steering committee member, and I'm a fresh career development fellow from JDRF, which started this year and will be for the next 5 years. And as Monica mentioned, the main two workhorse methods that I deal with is what we call hybridization capture, so virome capture sequencing, where we enrich viral sequences by next generation sequencing, and we use a comprehensive library of complementary oligo probes to kind of enrich for viral sequences prior to NGS. And then the other complementary technique is the FIB-seq, or also VerScan is an application of FIB-seq, where we are screening for antiviral antibody repertoire in all the longitudinal plasma samples. So yeah.
Well, fantastic. Congratulations on that fellowship. That's, I'm sure will be very helpful in getting this work done.
Yeah, and I saw Helena put in a cat photo. So these are two of my cats, and then my wife's a very big supporter of all the work. So
fantastic. I like the personal touch.
Thanks, Wook.
Take it away, Helena.
So today I'm going to give a little background of the ENDIA cohort study for those that don't know about us, and I'm going to talk mainly about the nested case control study, the why, the when, the who, and the what, introduce some omics data that ENDIA is collecting, and look at the data analysis approaches, and just give some final conclusions.
Fantastic.
So the ENDIA cohort study, ENDIA stands for Environmental Determinants of Islet Autoimmunity. So as the name suggests, we're trying to identify any environmental exposures or gene-environment interactions that trigger persistent islet autoimmunity, or IA. So persistent islet autoimmunity is our primary outcome, and it's defined as one or more islet autoantibodies. So that's to insulin, GAD, IA2 or the zinc transporter 8 on 2 consecutive occasions. In terms of recruitment, we commenced in 2013 and completed in 2019 with our first baby born in July 2020. And our intent is to follow children until 10 years of age. So recruitment was done in Australia, and we have a number of clinical sites and universities and research institutes participating. In terms of the ENDIA eligibility criteria, so these were unborn children during pregnancy or children who are less than 6 months old. And these children had to have a first degree relative with a type 1D proband. So this included mothers, fathers, and siblings. So these children have an increased risk of developing type 1 diabetes, which is around 16% higher than the children in the general population. So that's why we targeted this group in particular. So this slide represents 8 years of the ENDIA study. So our final infant population is 1473 from 1,453 pregnancies, and we have 1,214 families participating in ENDIA. So in terms of ENDIA follow-up, we follow the mothers during pregnancy and postnatally, and we see the children 3 monthly from pregnancy until the age of 2 years, and then 6 monthly thereafter until age 10. There are 4 key areas of follow-up. So health events, which include things like growth, illness, medical usage and mental health, lifestyle and physical environment, so family structure, physical activity, things like pet ownership, nutrition. So the parents and children have done diet surveys, and we also collect biospecimens. So in terms of our biological samples, we collect samples from the mother and the infant, so the mother during pregnancy and postnatally, and from the infant at every visit. And the biospecimens cover about 18 different types, from blood, stool, urine to breast milk. We also have some samples from the father and sibling probands. So the sampling is quite intensive. I think at the moment we have around 200,000 samples in our biobank.
Wow. Is that all stored at the University of Adelaide or?
It's stored at Adelaide. So there's a biobank facility at SAMRI where most of our samples are sitting. And obviously there's a few samples around the country where people are actually working on those samples.
Got it.
Okay, so why the nested case control study? So we know that there's high interest in environmental exposures during gestation and also maternal child transmission at birth that might trigger T1D. So the ENDIA study is actually the world's first and largest cohort study with recruitment during pregnancy. So we're positioned to offer a unique perspective on the significance of early life factors in the development of childhood type 1 diabetes, particularly obviously during pregnancy and the perinatal period. In terms of environmental exposures, ENDIA be characterizing various ohms. And for those of you that don't know what an ohm is, it's just the totality of a certain class of biological molecules, biomarkers, or analytes in a particular sample or cell type. For example, the microorganisms or microbiome found in your gut. So the main hypothesis in the nested case control study are that changes in the ohm are associated with our primary outcome of persistent islet autoimmunity, and that they reflect critical aspects of biology that underpin T1D risk. In terms of our aims, we're hoping to identify any differences in analytes or biomarkers between children with and without persistent IA. So these are our cases and controls. And we're going to do this at various time points, including pregnancy, birth, when the child develops IA and looking at the early childhood time trajectory. We're looking for both individual markers independently and also at interactions between biomarkers. So one of the main advantages of the NCC is that omics studies are very expensive, and because the NCC is a smaller subset of the cohort, it allows us to conduct the NCC and these omics studies where the cost would otherwise be prohibitive. So the NCC, the when or the why now. So the NCC was designed using the cohort case numbers at the end of 2019, and so we looked at a preliminary simulation in 2018, and 2019 was really where we projected that half of the cases would develop. So that was the thinking behind why it was 2019. So I guess the NCC is really an exploratory study. It's data-driven, discovery-focused, hypothesis generating, and we're looking at associations rather than causal relationships. So it's really a first look at the data. We recognise that we'll need validation of candidate biomarkers, and the plan is to do that looking at either the remaining cases that develop in ENDIA or validation from other cohorts, or possibly both of those. So what happens in the NCC? So we have our ENDIA cohort at the 31st of December 2019, and from that cohort we identified 54 cases, or 54 of our children who have developed persistent islet autoimmunity. And that's a combination of persistent single and persistent multiple antibodies, and within that we have some T1D cases. So in terms of our cases, what happens in the NCC, these cases are matched to controls, and they're matched on age and sex, and they also have to have negative antibody tests. The ratio of matching is 3:1, so every case has matched to three individuals in our study who we call controls, who have negative antibodies and are similar in terms of age and sex. So what happens in the nested case control in terms of matching, we use something called incidence density sampling. So for example, if we have a cohort with 5 participants with the same date of birth, and we follow them across time, we can see in this little example we have 3 participants, participant 1, 3, and 4, who have developed islet autoimmunity. In terms of choosing their match controls, if we look at participant 1 at time 1 when they develop islet autoimmunity, you can see that the potential in terms of other participants are those participants that haven't developed islet autoimmunity at that time. So that's 2, 3, 4, and 5. And similarly for 3 and 4, we can choose our match controls based on those individuals that haven't developed the disease at that time point. So the thing to notice is that participants can be both cases and controls. So a participant, for example, participant 3 can be a control at an earlier time point as long as they haven't developed islet autoimmunity. And also participants can be controls for more than one case. So in terms of our ENDIA study, we had 190 unique children. So some of our controls were controls to more than one case. We had 4 individuals that were controls and then developed the disease and became cases. And we had 50 cases which were only cases. So if you haven't tuned in so far, this is an important slide to catch you up. So this is our cohort at the 31st of December 2019. We have our cases which developed at that time, so our children that developed persistent islet autoimmunity. Each child is matched to 3 controls on age, sex, and negative antibodies. And these groups have matched controls and cases form risk sets. So the NCC is really a selected sample of the full cohort study. In terms of samples at birth and childhood, we have 10 different sampling types, up to 15 time points, depending on when the child develops IA, and around 12,000 unique biospecimens from these children that are undergoing various OMs. In terms of the mother, we have a similar number of sample types. We have 4 time points during pregnancy and postnatally, and around 3,000 unique biospecimens. We also have a lot of metadata. So we identified, along with the researchers, some priority variables for our NCC study. So in addition to the omics data that will be generated, we have around 1.6 million data points on these individuals and their mothers. So what are we doing in terms of the omics studies? These are the studies that are currently funded and are underway. So they range from the microbiome, proteome, fungal microbiome, the glycome, the lipidome, and obviously the plasma viral serology and the stool and respiratory virome, which Wook-Kim is leading. And just as a side thing, if you're interested in general omics study design, there's a section in the supplementary material of the paper that you might want to have a look at.
I just have a quick question here,
Yeah,
Helena.
sure.
Let's see, have you tried removing the cases from controls and comparing only controls and total cases? Are there any significant differences there between cases who developed versus cases who didn't develop earlier?
Yeah, so I guess we're not really powered to start diving down into starting to separate. In the protocol, we have said that we're going to do some sensitivity analysis, and that's something that we can look at as a sensitivity analysis. So the answer is no, but in terms of our primary analyses and secondary analyses, but it's something that you can look at in a sensitivity analysis.
Yeah,
Okay.
I think it's coming from, because as a researcher, it's we had many debates and discussions about this, how can a case be considered a control at a time point when it's just simply not a case, one said. But after many discussions, we agreed on this design, and then agreed that if we can, we would do sensitivity analysis. So I think, yeah, we know where you're coming from, and that's something that we definitely want to explore, and as well as we actually analyze as a case cohort design and analyze all the case, then we will have sufficient power to kind of address this question. Yeah.
Great question from Kiati in Boston.
Yeah, I think also in the protocol we said that we will include for each individual, regardless of whether it's a case or control, within that particular risk set, we will include all that information within that risk set, and then the sensitivity analysis will be taking out maybe those individuals who cross over from cases or to controls. So yeah, as Wook said, there was a lot of debate on what was finally put in the protocol, but I guess our final decisions are what we really need to kind of stand by, because
yeah, I mean, you have
there
to
was
start
no point in publishing the protocol otherwise.
Yeah.
We're not going to.
You've got to start somewhere. So yeah,
That's right.
That's great. Okay, thanks for answering that.
Okay, so in terms of the omics data in the nested case control study, we have longitudinal data, so data over time. We have our IA status, so whether the child is a case or control. We have different omic platforms, different sample types, and obviously mother and infant data. In terms of our data analysis, so in the protocol paper, we really just looked at how we would analyze each omics platform separately, and also sample types separately. So our primary analysis is a weighted conditional logistic regression. So this is when we're looking at a single time point. So in this case, the onset of Islet autoimmunity, and looking at a single analyte or biomarker at a time. And obviously there's various combinations of time, and looking at analytes in a multivariate way. And these are, I guess, the methods that we finally decided on. These other methods, we've called them secondary analyses, as I guess with the nested case control study, the way it's designed, it is really for a single time and a single biomarker. So that's the weighted conditional logistic regression is our primary analysis. So what we don't talk about in the protocol is how we might integrate the omics analysis. That's really quite a complicated procedure, and at the moment there's no methods that really can deal with the type of data that we have in a good way, I guess. So the fact that we have got longitudinal data, we've got disease state, IA status, and we've got all these different platforms from different sample types. So that's an area for hopefully future research and funding. So final conclusions of our ENDIA nested case control study. So we've got longitudinal omics profiles, we've got mother-child dyads, we've got omic platforms and sample types, and hopefully hundreds to thousands of analytes. So the holistic approach of the NCC will enable collective identification, quantification of key omic analytes, and hopefully provide an opportunity to explore how they change over time and how these biomarkers interact at different stages of the life course. And I guess that our main goal is to enable through these studies the development origins of type 1D to be explored. So obviously I'd like to thank our funders, the researchers, and a big shout out to all our ENDIA families who come in every 3 to 6 months and have lots of samples taken from them and fill out lots of surveys for us. You know, without them, we wouldn't have our study. So thank you.
Well, thank you very much, and thank you for all your hard work in this area. I have a couple questions. You know, in terms of accessing these data, are these data just available to the Australian institutions and academics, or, you know, if you're in Europe and Canada, the US, are you able to petition for access to like raw samples or actual just the data generated? Typically the listener here, so you know, and they may be intrigued by this mass amount of data.
So I guess generally what we would like you to do is send an initial email to I think probably you send it to my email, or we have an ENDIA at adelaide.edu.au email. Any requests for data or samples, generally we have to fill out kind of, I guess, an overall view of what you're proposing, and that would go to our steering committee. Obviously, we're still in the early stages and we're still collecting data, so we've got a few more years to go. I think the study, our last child was born in 2020 and we're following them up till the age 10, so the study's not due to finish until 2030. So but yes, you can ask for access to data and samples.
Okay. What is that
Yeah,
timeline for that process usually is it like weeks, months?
It's, I think, I guess I have to be honest that it usually is months, because we have to, we meet steering committee each month. Usually what we would ask is to fill out a form, but also maybe present if we think is appropriate, and then get an idea of what the data will be used for. But having said that, there was a recent RFA call that was organized by JDRF Australia, and we're reviewing applications now. So that process has closed now, and then that was exactly the purpose of the RFA, was to have external collaborators wanting access to samples or data or both. And the requirement was that an internal investigator, so an ENDIA investigator, had to be part of that research team. So that's already in process and being peer reviewed, or have just finished peer reviewing.
We promoted that effort, and that's a great collaborative effort that you guys put together, and we're reaching out into the global community to partner with. So kudos. And that'll be very interesting to see what comes out of that, what kind of projects come out of that.
Yeah.
In terms of the microbiome projects.
Oh, sorry. You go.
Sorry. Yeah. In terms of the microbiome projects, just because we have some representative of a lab that has interest in the microbiome, who's heading up that that project or projects that are related to the microbiome? I know you're doing the omics.
Yeah,
So that would probably be
Harrison, is leading the bacterial and the fungal microbiome, and then
That's Emma, you said.
And then Emma as well, Emma Hamilton-Williams. She's looking at meta proteomics, which also is looking at human and bacterial genes or proteins in the stool.
Yeah, we've been following her work for some time now, and it's excellent. And then I see, and Len Harrison. We heard his talk. I think we heard his talk at IDS. So I think this is a really interesting data set that you've developed and are building. And I love that you're sort of flying in, building the plane while it's being built. You're starting to really start inquiry, and it's really exciting to see what will come out of this. As you said, this is sort of the first of its kind. So it'll be, there'll probably be some learning curves, but also we expect, and we've heard from many people, that many scientists, that this is going to be a very important resource for the T1D scientific community. Shout out to you both, and I hope that everyone takes a peek at the paper. Do you think that, one last question, do you think they'll put out another RFA following this one, like maybe on a yearly basis or something like that?
I'm not sure.
I'm not really sure, but I don't think this is a one-off, and I think we've had internal discussions, and Helena and the data team have worked to get sort of external consultants to kind of see whether we should be setting up a repository of some kind, something that would benefit, I guess, even the internal investigators. We want to make sure that all the data is sort of centralized for, forever access, rather than, if a particular investigator team moves on, then, what happens to that data. So we are trying to centralize that. And
there it is. Thank you. Thanks for the link in the chat. I would say we've heard a lot about that recently, as we initiated, after we held a data-driven challenge, and after some of the participants gave us some feedback about the paucity of TCR data, we initiated and facilitated the build of a T1D focused TCR database. And so there's just a lot of moving parts in doing that, particularly with the scale of data that you're capturing. I think it's a massive and monumental effort, and so just really in awe of what you've got going here. Okay, well, thank you both very much, and have a great rest of the day. And I'm hoping that this gets on the radar of those that it's not on. And we're fingers crossed that you start pulling some very interesting data out it soon.
Thank you for having us.
Thanks
It's been
very
a
much.
pleasure.
Thanks.
Thank you.
Thank you, and have a great rest of your day. Bye bye now.
You too.
Bye.